All right, so good morning, everyone, and welcome to our seventh webinar in the series of ticks and tick-borne pathogens open community webinar series. My name is Anna Maria Niewiadomska, and I work for the viral side of the bacterial and viral bioinformatics resource centers. And today I'll be talking to you about comparative genomics of Flaviviridae in ticks. I'll just go over the logistics of this webinar just very quickly so that we can give other participants a chance to join in. And I apologize for my voice this morning. It's a little bit croaky, getting over a cold, but hopefully it holds out till the end of the webinar. So this is being recorded and will be available on the vupathdb.org website at this link, this address over here. As I said, this is part of an entire webinar series on ticks and their pathogens, and that includes eukaryotic pathogens, the vectors that transmit them, viruses, and bacterial pathogens that are transmitted by ticks. We have one more webinar left in the series in addition to today's, and that will be on June the 14th on RNA-seq and Snip analysis of bacteria transmitted by ticks, and that will be presented by my colleague Rebecca. And if you haven't registered for that, you can go ahead and do that there. And if you'd like to watch any of the other previous webinars, you can find them on this page as well. So today I thought I would just give you a quick overview of the BV-BRC and the viruses that we'll be talking about. And then I thought we could move on to the website itself and go through some use cases. And hopefully we'll have a little bit of time left at the end for some Q&A and discussion. So just real quick, if you're not already familiar with the BV-BRC or the Bioinformatic Resource Centers, we've been around for quite a while, but our sites have taken different forms. Most recently, we've merged several of our databases into two databases, the eukaryotic pathogen database and the vector database have merged into VUPathDB, and this hosts data on eukaryotic pathogens and their vectors. On the other hand, we are part of the BV-BRC, and the new website, if you're not already familiar with it, is a joint production and a merging of the previous sites, which include the Patrick database, which focuses on bacterial pathogens, and the IRD, the Influenza Research Database, and the Virus Pathogen Resource, which have both historically been focused on viral pathogens. You can find all of the same information in addition to new viral groups and new tools at our new website at bvbrc.org. So if you're wondering what kind of tick-borne pathogens you can find, there's quite a few bacterial tick-borne pathogens, and my colleague Rebecca has gone over some of these. And there's quite a few of these, and you can go ahead and browse them and explore them on our site. My focus, however, is on viral pathogens, and we focus on several viral families that span the different types of viruses, such as single-stranded RNA viruses and double-stranded DNA viruses, things like that. And from these, we have several viral families that are transmitted by ticks. My previous webinar was on the Flavivirus order, which has multiple viral families transmitted by ticks. And you can watch that, as I said, on the website. But today I thought I would talk about flaviviruses since they're quite different. And in particular, I'll be talking about a use case with Tick-Borne Encephalitis virus. And there's quite a few genome segments, so there's a lot of data to explore. And I would encourage you to do that yourself if you're interested in other tick-borne viral families. So I thought I would just give you a quick introduction to the Flavivirus family. These are RNA viruses that mainly infect mammals and birds. They're primarily spread by arthropod vectors, and you're probably familiar with, and most people are probably familiar with those spread by mosquitoes such as dengue virus, yellow fever virus, but tick-borne flaviviruses are less well known, but still quite important both to humans and agriculturally. The Flaviviridae family is divided into 4 genera: Flavivirus, Hepacivirus, Pegivirus, and Pestivirus. But the majority of the tick-borne viruses belong to the Flavivirus genus. Only one of them can be found in the Hepacivirus genus, which mainly has hepatitis C virus in it. There are a few unclassified tick-borne viruses that still haven't been assigned a genus, but most of these are related to the Flavivirus genus. So I thought I would give you just a quick list of what species in the Flavivirus genus are known to be transmitted by ticks. And these include a lot of viruses with very interesting names like Gadget Scully virus, Greek goat encephalitis virus, Chiastinor forest disease virus, but probably the most studied and most well-known are Powassan virus, which can be found here in North America and some parts of Asia, and Tick-Borne Encephalitis virus. And all of these are somewhat related to each other phylogenetically, but we'll get into that in a little bit more detail later. So if you're interested in what kind of ticks carry these flaviviruses, it's more than just one specific species, and they can be divided into softback ticks in the Ornithodorus species and hardbacked ticks. And multiple types of ticks can transmit the same virus. So you can see, for example, al-Humra hemorrhagic fever can be transmitted by both soft and hardback ticks. But one of the most studied species is, some of the species that are most studied are in the Ixodes family, and these can transmit a great many viruses. And here's a nice picture of a cute little sheep tick called Ixodes rhynchus, and that happens to transmit tick-borne encephalitis virus, which I'll be focused on today. So just to give you a little bit of an idea on the characteristics of these flaviviruses themselves, as I mentioned, they're positive sense RNA viruses. They have a single strand. The viruses themselves are quite small. They're only about 40 to 60 nanometers in diameter, and they are enveloped viruses. The virus genome itself is also quite short and compact. It's only about 10 to 11 kb, depending on the species. They have a 5' cap, and all the proteins are encoded in a single open reading frame with 3' and 5' UTRs. And this open reading frame actually encodes 10 viral proteins, 3 of them which are structural proteins that make up the basic parts of the virus, including the envelope, membrane, and capsid protein. And on the inside, we have the genomic RNA with some nice conserved structures on the 5' and 3' ends. And we also have 7 non-structural proteins that have different functions, including genome replication and immunomodulatory functions. So this is Just to give you a quick idea of what the genome of this virus looks like. And this is the one long open reading frame that we talked about, and it makes just one single long polyprotein. But this polyprotein can then be cleaved into multiple different proteins, or mature peptides, as they're called. So here we've got our structural proteins, the capsid, membrane, and the envelope proteins. And then we have all the non-structural proteins that start with NS, like NS1, NS2A, and so on and so forth. And this is just a quick overview of the phylogeny of these tick-borne flaviviruses. And as I mentioned, they're quite related to each other, but it also turns out that they cluster in terms of their genomic similarity, depending on the vector that they're transmitted by. So over here, we have this nice cluster in red, and these are our tick-borne virus complex. And these two down here are transmitted by seabird tick-borne viruses. So these are restricted to birds and typically cause disease in birds, whereas these infect mammals and occasionally they jump from animals to humans. And down here we have our mosquito transmitted viruses, which many of you are probably more familiar with, such as dengue virus and yellow fever virus and Zika. So I'll be talking today about Tick-Borne Encephalitis virus. Generally, when people are infected with this virus as a result of being bitten by a tick, symptoms are typically mild and some people are even asymptomatic, but occasionally you end up with infection of cells in the central nervous system, and this can cause severe disease such as encephalitis, meningitis, and it can cause other neurological symptoms and issues in patients. There's not many treatment methods out there, and usually people are just given supportive treatment. However, people have been experimenting with vaccines for a very long time, particularly in Europe and in the Soviet Union historically, and now Russia. And there have been many different types of vaccines that have been developed. But, and in 2021, the FDA approved one of these vaccines for use in the United States. So Tick-Borne Encephalitis virus has quite a long history. It's been known about for almost 100 years so far. And about 20 years ago, people believed that, or what was known about tick-borne virus, based on how it clustered in a phylogenetic tree, was that there are about three different groups, or three main groups of tick-borne encephalitis virus, one of which was the European group that mostly was found in Europe, a Siberian group mostly found in Siberia, and Far Eastern group, which was found in some Far East Asian countries as well as Eastern Russia. But since then, as this virus has grown and as surveillance for this virus has grown, and particularly with climate warming, it's become more of a problem, and with the spread of ticks into more geographic places, surveillance has increased, and several new divergent tick-borne encephalitis virus strains have been found that fall outside of these three main groups. For example, there were two divergent tick-borne encephalitis viruses found in the Irkutsk region in 2016 in the Netherlands. A new lineage of, or a new European subtype was found. And in 2018, a very divergent subtype was found in the Tibetan highlands that didn't cluster with any of these three groups on the phylogenetic tree. So I thought today we could go to the BV-BRC website, take our time. I could show you how to look for specific data set that you might be interested in exploring. We'll then, once we have our data set, we can use that to generate multiple sequence alignments. And this will be using our multiple sequence alignment and Snip Analysis tool. After that, we can use our multiple sequence alignment to generate a phylogenetic tree and explore for ourselves what the latest data and what the latest genomes in the Tick-Borne Encephalitis virus species look like. See if we can see any kind of geographic clustering. And we'll explore the phylogenetic tree builder tool to use this, and our Archaeopteryx viewer to look at and explore our tree. And then finally, we'll try to compare variations in a specific protein, for example, from these three phylogenetic groups that I mentioned, the European, Siberian, and Far Eastern groups. And this will showcase our Metacats tool. So I will check if there's any questions, and if not, we can move on to the website demonstration. All right, looks like we're good. So let's just maximize the screen and make sure everybody can see.
Looks good. Yep.
All right, fantastic. So if you haven't used the BV-BRC.org website before, it's pretty easy to get to bv-brc.org. As I mentioned, you can find all kinds of data and information on both bacteria and viruses. And we do also provide information on archaea, but that's not today's topic. So there's many different types of bioinformatic data, and it's not just restricted to genomes and protein data, but you can also find metabolic data, protein structures, and a plethora of other things. And you can find that by just clicking on the specific pathogen you're interested in. And for me, that's viruses. And this will take you to the virus landing page. And this gives you just a quick idea of what you can find in here in terms of genomic data. It tells you how many viral families we have, how many genomes or genome segments, proteins, protein structures, and they're divided, of course, into the different types of Baltimore group families. So over here, we have our single-stranded positive sense RNA, which is where I will find the Flaviviridae family. So I'm going to go ahead and click on that. And this takes me to the Flaviviridae landing page. And if you're lost, you can kind of just look up here, and the breadcrumbs will give you an idea of where you are in terms of the viral taxonomy, because this is the taxon view. But you can also flip through the different tabs and look at the different information that's associated with this virus family. So we've got taxonomy, if you want to browse through the different viral species that way. We've got all the different genomes. We've got the proteins, protein structures, domains and motifs, epitopes, if you're interested in the immunological data. And we also do have some experimental data for some, but not all of the viral families. If there's something you're more specifically interested in, you can also just type it into this quick search box. And you can either leave it to the default all data types, and that will give you what you want. So for example, I could just do Tick-Borne Encephalitis. And that will show me what's in the database. So it found 2 matches underneath the taxa. One of them is Tick-Borne Encephalitis virus, which is what I want. And one of them is chimeric Tick-Borne Encephalitis virus, or dengue virus 4, which is not what I'm interested in. So that's how you can find the data that you're interested in. And this takes me, again, if we follow the breadcrumbs, we can see the taxonomy of it. So we have the Flaviviridae family, the Flavivirus genus, and then over here, the viral species that we're interested in, Tick-Borne Encephalitis virus. So now I'm going to flip to the genomes tab, which will show me a list of all of the genomes that are available in the website. So this is quite a long list, and there's a lot to take in here, but it's actually very easy to navigate through the data because all of the metadata that we import from GenBank is here. And in addition, we also add our own curation onto it so that we can standardize and normalize the data, making it more easy for users to search so that they can search by geographic group, for example, and you can get to that by clicking on the filters tab. And this will give you a way to just start putting together your data set based on the metadata that you're interested in. You can choose by whether or not you're interested in complete genomes or partial genomes. So for my case, for my use case, I only want all of the complete genomes that are there, and that's 277. You can choose the specific country that you're interested in. If you're only interested in isolates from a specific country, a geographic group, Asia or Europe, which is where this virus is most widespread. If you're looking to study the virus based on a specific host, again, you can choose this. And let's say you only want viruses that have historically infected humans. And if you're interested in other things as well, you can explore by clicking on this gear button, the other fields that we have available, and there's quite a long list. And so once you've selected what you want, you can also customize how you view the metadata in this table by clicking on this plus button on the top right corner over here. And it's actually quite an impressive list so that you can customize the metadata that you want to see and customize the metadata that you want to download as well. So if you just scroll down, for example, let's say I'd like to see the scientific name of the host that these viruses are transmitted in. I would scroll down to the host info subsection and click on the host name. And so this adds the host name that's provided in GenBank for each one of these. And we also have the host common name. And another thing that we do is the host group. So let's say, for example, I'm interested in whether or not these particular viruses were isolated in ticks or humans. I might not want to scroll through the entire list of scientific names and figure out whether Ixodes rhynchus and another type of scientific name are both ticks. I can just go to host group and select just ticks. So this is quite useful, and I find that it helps save time. So let's say, for example, I'm interested in all of the Tick-Borne Encephalitis viruses that have been collected in the last 20 years, because it seems that the more divergent Tick-Borne Encephalitis viruses have mostly been newer isolates. So I can click on, let's say, a particular collection year, or I can use the advanced search button to click on a specific metadata title that I'm interested in. So for example, let's see where it would be, the host collection year, and specify that I'm only interested in viruses from the year 2000 to the year 2022 and click on search. And so that gives me a list of 115 results. And you can find out more about the individual isolate by just clicking on it. And this populates this, this tab on the right, where you can see all of the information that's either been imported from GenBank or added by our own curators to the database. And it's divided into several subsections, for example, taxonomy, strain information, whether there's any information on the sequencing platform, which can be relevant for people doing particular kinds of studies, whether or not it's been annotated or not, and more information on how it was isolated, when it was isolated, where, and of course, the host information subsection. So if you'd like to select more than one of these, you can, of course, go through it just by clicking one or the other, or you can hold down the shift button, which I found quite useful, and just scroll down and click on as many as you want. All of these columns are sortable. So let's say, for example, you want to order it by collection year. You can just go ahead and click on that. And of course, if you want to widen the table, you can click on this hide button to toggle the details panel out of the way. And again, as I mentioned, you can customize this metadata table and add or hide as many columns as you're interested in. For our use case, I'm going to select all of these genomes and go ahead and group these, because I want to save them as a group and be able to align them and then make a tree out of them eventually. So I can click on group button. And so this gives me a little pop-up that shows and gives me the option to either save it to an existing group or save it to a new group. And then another thing pops up with this little file icon, and this allows me to select where I want this information saved. Every user account has a default folder for genome groups, and these are basically just a collection of all of the genomic or nucleic acid sequences that you want to keep together for later analysis. So in my case, I've made a specific folder called Flaviviruses and Ticks, and I'm going to go ahead and click on that, and we can call this anything that we want. So let's say Tick-Borne Encephalitis virus, genomes 1. And you can go ahead and click on add. And so this will take a couple of seconds, but down here you'll get a little pop-up that tells you your group was created. We also have another option, another type of data set called a feature group. And this is usually how you would want to save the proteins rather than the nucleic acids. So if I switch to the protein tab, this will show me the different proteins that are associated with the genomes that I had previously selected. Oh, and one more important thing I forgot to show you. So now that we've selected that we only want complete genomes and that we only want them for a specific date range, if you want to see the proteins only for this subselection, you do have to click the apply button. Otherwise, it will show you the proteins from the entire Tick-Borne Encephalitis virus collection. So now when I switch to the protein tab, instead of showing me all of the results, it's going to be only 14 proteins. And the annotation also depends on whether you're looking at the internal annotation, which right now is called Patrick, or whether you want the annotation from that was imported from GenBank under the RefSeq tab. And then when you look at the feature type column, different things can be annotated, including RNA elements, as well as proteins, which are coding sequence features and mature peptides. So not only can you find information on that one long polyprotein chunk, but we also use peptides prediction tools to give you an idea of where exactly these smaller mature peptides that have been cleaved from that polyprotein are. And so let's say I want to save a subsection of these. I can go ahead and click on those. And I can also click group over here. But in this case, it's going to tell me that I'm saving it as a feature group, which would include the proteins rather than the genomic sequence. So I hope that gives you an idea of how to browse for the different kinds of information that you can find. And I would encourage you to also explore these other tabs that contain information on protein structures, specific different structural domains and conserved motifs that you might find, specific immune epitopes if you're interested in those, and so on. But next, I'd like to move into how you can analyze this data now that you have it. So you would go ahead and click on Tools and Services, and you'll find a list of the different tools here that you can use to analyze your data. And because we do have specific tools for analysis of bacteria and specific tools for analysis of viruses, you might want to pay attention to this little B or V that's found after each tool. And the B specifies a tool that might be mainly used for bacteria. In some cases, it can be adapted to viruses, but initially it was optimized for bacterial. And then, for example, we have our SARS-CoV-2 genome and assembly annotation tool, and obviously this one would be restricted only to viruses. But things like a multiple sequence alignment tool is something that you can use for both bacteria and viruses. So that has no letters after that. So that just gives you a little bit of a hint on how to kind of navigate through the tools and services on the site. And at the top of each page, you'll find a little summary explaining what this tool is about. So if you want to browse through the different tools and just explore what they are, you can go ahead and do that. And for each one, we also usually have a quick reference guide to just give you a quick idea of what these buttons and different parameters are that you might have to select. And it's really useful if you're not familiar with the terminology of the new site, especially if you're coming from the Viper side of the website. And you can also find a link for a tutorial that will help walk you through in more detail how to use a specific tool. So this service combines a couple of different things. One of them is just making a multiple sequence alignment. So you could start with unaligned sequences, but it also looks at and gives you an idea of the variation or single nucleotide polymorphisms that you can also find in a multiple sequence alignment. So you can also, if you already have a multiple sequence alignment on your own computer, upload that over here and just use that for a SNP analysis. And then you have different types of data that you can input. You can select a feature group if you're interested in uploading protein information, or you can select a viral genome group if you want to input the entire viral nucleic acid genome, which is what I'd like to do. So to find that, you can either start typing. So I would put TBEV, but that's not going to come up. So sometimes you might have to look for your data by clicking on your folder and then navigate to the specific subfolder that you created. And in my case, I have my Flavivirus and ticks folder that I created specifically for this webinar. And over here, I have my genomes, which are the Tick-Borne Encephalitis virus from 2000 to 2022. And in addition to these, I also added one more genome that was an outgroup so that I can just have a better idea of what's there. So I can click on OK if I'm happy with that. But if you also want to upload your own information, you can. You don't have to stick to information from the website. You can also click on the upload button and then just select your file or just drag it into this box to upload that. If you're looking to create a specific folder and then upload, you can use this button. But again, we already have the file here already ready and prepared. So I'm just going to go ahead and click on OK. You can choose the specific kind of aligner that you want. My preference today is using the mapped aligner, but we also have the muscle aligner available. And then the last thing you have to do is just write down or input the information for where you want the output files from this job generated. So again, I can go ahead and click on my Flaviviruses and ticks folder, and then just give this a name. For example, multiple sequence alignment, Tick-Borne Encephalitis virus. And then once all of the information is there, this grayed out submit button should become available to click, and you can go ahead and click submit. If everything is okay, it should tell you that your job has been submitted successfully and that you should check the job list to find out when or how your job is done. So you might think, okay, how do I find out where my job is now? So if you're not familiar with this site, there's a couple places you can go to do that. You can click on workspaces and click on my jobs. Or if you've been paying attention, you'll see this little box down here, and you'll notice that that changed as soon as I clicked the button. So this gives you, these two boxes give you a summary of what's happening on the site. This tells you the status of the files that you're uploading. And if you've uploaded something, it'll tell you whether it's still uploading or whether it's been successfully uploaded. And over here, it'll give you an idea of how many jobs that you have that are either queued in gray, that are running in yellow, or that have completed in green. And if you just go ahead and click on that, that takes you to a list of your job statuses. And you can go ahead and click on the one that you're interested in to see your results. So this gives you quite a long list of files sometimes. And again, if you're not familiar with these file extension names, you can definitely go through our reference guide and tutorials so that you can understand what these different output files are. But we also understand that people just want to see their results. And most people who want multiple sequence alignment know what a multiple sequence alignment looks like. So you can just go ahead and click on this quick view icon to view your multiple sequence alignments. This opens up a new tab over here, which launches our multiple sequence alignment viewer. And you can scroll up or down. You can click on a specific sequence that you're interested in, and you can move this around to kind of get an idea of how good your alignment was. So for example, at the beginning of the alignment, we have several genomes that are missing the first 100 or 150 nucleotides. So you might want to trim this alignment before you move further on with your analysis. Over here, we have a few different buttons you can use to change the color scheme of these of this alignment. And I would again encourage you to explore these because there's quite a few interesting things over here. For example, you can look at different structural aspects of these nucleic acids. You can also use filters to show different columns depending on whether or not you're interested in columns that do or don't have gaps in them, or you just want to look at conserved sequences. And again, you should, if you have some time, just play around with those. And you can also switch the ID type. So if you look over here, these names might not make that much sense to you, and that's understandable. These are our internal database names, but you can switch these to anything that you want, whether it's the genome name, for example, or the accession, or the species, or the strain. And that sometimes helps to really give you an idea of what types of viruses are maybe transmitted by a specific host or specific in a specific geographical area. So again, I also encourage you to play around with those. And finally, you can also play around with the different visualization options that show you the conserved weight, for example, of each single position, and so on. And then if you just want to download that, you can either save a particular image by clicking the download button over there. Let's say you're interested, for example, in this particular residue over here in this block of Gs and how many sequences have that. You can go ahead and do that. But going back to the list of the files, one other thing I'd like to point out is the Snip Analysis. And that's the snip.tsv file over here. And you can find that or view that by clicking on it and then clicking on the view button over here to see it in the browser. You can also download it and open it up in Excel to see what's there. But this will give you an idea of what's found at each position. It will give everything a score. It will show you the consensus of each position and the number of sequences that contain that particular residue over there. So now that I have my multiple sequence alignment, the next thing I wanted to do was to make a phylogenetic tree out of this. But it's not necessary for you to go through the alignment step first. You can just go straight to making a gene tree, and this tool will make the alignment for you, then generate the tree. So again, over here, you have the option of choosing whether you want your input to be DNA or protein. And as I mentioned, you can add aligned sequences or you can have unaligned sequences. And if you already have your data in the database or saved as a feature group of proteins or a genome group, you can go ahead and add that over here. And so in this case, again, I'm going to choose my Tick-Borne Encephalitis viruses from 2000 to 2022 and click on OK. And it's important that you click on this plus button so that it gets added to the feature or file table that can be aligned. Over here, you can specify the alignment parameters. And because I noticed that, let's say maybe the first 100 or so bits of, or the first 100 or so nucleotides of my alignments were missing in some genomes, I'm going to go ahead and click on 0.1% to trim the first 10% ends of my alignments. And you can also choose whether or not you'd like to remove specific columns that have more than a specific percentage of gaps. So again, I'm also going to click on 0.1%. And then you can choose what kind of tree you'd like to build. So over here, we offer 2 maximum likelihood trees, RAXML or PhyML. But since I'm in a bit of a rush today, I'm just going to click on Fast Tree, which kind of will just give you a quick overview of the structure of your tree. I would recommend that you use RAXML or PhyML for if you're trying to generate a tree that's for publications. You can also choose your specific substitution model. over here if you would like to do that. And again, the last thing that you do is just specify the output folder. And so I've got my Flavivirus and ticks folder, and then I'll put BV tree and click on submit. Again, it tells me that it's creating the tree and it's been successfully submitted. I can go down over here and see that the job is running and it's been completed. But because this takes a little bit of time, I'm going to go ahead and click on a pre-computed tree that I made yesterday so that we can take a look at that. And so I can find what I'm looking for based on the type of tree, the type of service that I made. And over here, it's the gene tree. And I'll go ahead and click on that. And again, it gives me a list of different output files that I can explore. But if I'm just interested in taking a quick look at the tree, I can again go up to the top right here and click on this view button, and this will launch the tree viewer. So it might take a couple of seconds to load, but be patient, it usually does. So here we have our tree starting to load. And sometimes it can be a little bit glitchy, but we're working on that. So you can just refresh the page. It's taking a little while. And here is our beautiful tree. So if this tree viewer is new to you, it can take a little time to learn all of the different buttons and options that are available. But I would encourage you to explore it because it is actually a really rich tool that gives you so many different options for not only viewing this tree, but manipulating it with different colors and different shapes so that you can really find the perfect tree for your publication, or to help you understand or help other people understand what it is that you're looking at. So there's a lot to take in here, but if you look on the left side, you can change or customize the information that's displayed here. So I can unclick node name, and that takes off the internal ID. You can choose whether or not you want to view the collection year, and that's something that I'm always interested in, to see if there's any correlation with time. So you can click on collection year, and that shows you the year the virus was isolated. You can click on geographic group, and that will show you whether it was isolated from Europe or Asia. Now, that's not that interesting to me. I'd rather look at which country it was isolated from. So I can click on that, and that populates Russia over here for me. But I'm finding this information a little bit hard to see over here. So I'm going to take off the confidence level for that. And that helps me see the structure of the tree and the individual branching a little bit more. So if I want to zoom in on a specific branch, I can also use the zoom buttons to either zoom in vertically, which will kind of expand the compressed branches of the tree, or I can zoom in horizontally, which also helps me expand it if I need to do it that way. And if I want to go back and reset the tree to the initial view, I can go ahead and click on Fit and Center. And if you're not sure what any of these buttons do, you can just hover over them and it'll tell you exactly what their function is. So I've gone ahead and reset that. You can change the label size if you're having trouble seeing it. You can change the node size, you can change the branch width. It's really nice to be able to have all of these options to customize it. But one of the things that I like most about this tree viewer is that you can colorize it depending on the metadata that's available in the websites. So for example, we've got the collection year that you can color by, the geographic group, the host group, the isolation country, the species. And because from what we know about Tick-Borne Encephalitis virus, we know that the main groups are dependent on the countries that they're from. I'm going to color it by isolation country. And so this really helps us visualize those three main branches that we first discussed when we were talking about Tick-Borne Encephalitis virus. Down here, towards the bottom of the tree, we have our European group. And we can see that most of the samples do, in fact, come from European countries. We've got Denmark, the UK, Finland, Germany, Hungary. And then over here, we can see our, our Siberian group, most of which come from Russia and, or at least Siberian areas in Russia. And then up here, we've got our Far Eastern group, which includes species from Russia and China. And then we also have those few groups, those few divergent groups that we talked about that don't fall in between any of these well-defined groups, such as these two from Russia over here. This one that was isolated from Tibet in 2013, or the Tibetan Highlands. And then we've got this strange one from Austria, and a couple other more divergent ones down here. If you want to zoom into a particular node, you can click on that node and you can either collapse it if you're not interested in it, or you can go to the subtree to zoom in on it and look at the branches in more detail. So you can see, for example, that there's a particular cluster that is restricted in this group mainly to Russia, whereas these are more commonly found in China. And again, you can download your trees as pictures, as Newick trees, as phyllo-XML trees, and use these for your publications. And so finally, the last thing that I wanted to show you real quick, and I'll try and get this done so we still have some time for questions, is our Metacats tool. And this one is a metadata-driven comparative analysis tool. And what this does is helps you look at whether there are positions either in the nucleotide or a protein that differ significantly based on either groups that you've chosen or groups that are auto-grouped based on the metadata that's available in the database, such as the geographic location, the specific country, or when the sample was collected. So I thought it might be interesting for us to compare whether there's any specific differences in the envelope protein of these three different groups, the Siberian, Far Eastern, and the European. So I went ahead and pre-assembled feature groups for these three different subgroups of Tick-Borne Encephalitis virus. And using our protein search, I collected just the envelope protein for those. So I'm going to go ahead and put our output folder as our Flavivirus and ticks, and put Metacats demo. And because I already know and I've already made my own feature groups based on those three particular Tick-Borne encephalitis groups, I'm going to go ahead to the folder where I saved them and click on group 1, which is the Far Eastern group envelope proteins. Add that. Go ahead and click on the Siberian one. Add that. And finally, the European envelope proteins, and always remember to click the plus button to add it. So now I have my 3 groups, and what this tool is going to do is compare them and identify any amino acid sites that are significantly different between the 3 groups. So you can go ahead and click on submit. And then again, just in the interest of time, I've already pre-calculated this. So I'm going to go ahead and click on that. This gives me a couple of different output tables showing the statistical significance of each site. I'm going to click on first row contains the column headers to populate this table. And this shows me every single position for the envelope protein, which goes from, it's quite a short protein. It's only about 200 amino acids. It shows me the chi-square value, the p-value, so anything over 0.5, which is what I set in the parameters, would be considered significant. And over here, it tells me whether or not it was above or below that threshold. Shows the degrees of freedom. And again, I would encourage you to read the documentation to understand what each one of these are. And these are my 3 different groups over here, but it can be tedious to just scroll through these. If I'm only interested in just the significant positions, I can go ahead to the keywords over here and use, click on significant and say that I only want the ones that have a Y or a yes for significance and filter those. And this shows me the positions. So for example, at position 88 in that protein, I've got in one of these groups, most of them have a serine, whereas in this particular group, most of them would have a G in that position. And you can use these to develop diagnostic PCR primers, to do immunological studies, to find specific epitopes that you might be interested in exploring. And there's many different applications to using these tools. And it's a really useful and very powerful way of finding what really defines a particular protein or nucleic acid from a specific group. So I will stop there because we're almost at the end of the hour and open it up for questions.
That was really nice, Anna. Thank you.
Thank you all for your attention.
I can start off with a question maybe until people get warmed up if they have questions. I
No
just
problem.
quickly, I'm sure this came up in a previous webinar, but how many sequences can you actually align? Is there a limitation to that, and or is the sky the limit?
Right. So it really depends because bacterial and viral pathogens differ so much in terms of their size. I mean, you have a 10 kb RNA virus, or you have, it's a 400,000 base pair plasmid. It's not going to be the same in terms of your input data size. So we're really trying to find the sweet spot and kind of customize it for each species. So it really depends, and we're still working on it. But we're trying to find a solution that makes sense for every species.
Right. Yep.
So we won't allow you to, for example, align whole bacterial genomes, but you can align whole viral genomes. Okay. So in the meantime, I will, while we're seeing if we have any more questions, I'll just mention that if you want to find out more, we do have a lot of instructional videos on YouTube showing you kind of how to just navigate through the sites and the different tools. We had a webinar series kind of introducing the BV-BRC. We do have a lot of written tutorials and guides. We also have a help desk. And lastly, if you want to just keep up with us and what we do, you can follow us on social media. This is our Twitter handle, where you can find us on Facebook. And if you just have any general questions or want to discuss things relating to the BV-BRC or at all the BRCs actually, You can also find us on our subreddit, BRC users, and just start a discussion. And if you're having trouble with the site, you can go to our About tab and click on Contact Us. That opens up a pop-up and you can say, you know, hey, I love your site, thank you so much. Or, you know, hey, I don't like your site. Where's the old one? Or, you know, I'd like to help you improve the site and give us a suggestion. Or, you know, I think this particular data type would be really useful for you to add. So, you know, please feel free to send us your feedback. We really try to gear this towards our users, and we do listen to what you say. So please do share with us if you have a problem or if there's any feature that you particularly like or dislike, or think you can help us improve anything. All right. Well, if there are no more questions, then I just want to thank you all so much for showing up and listening. And I appreciate your attention. And again, if you missed anything or would like to see this again, this has been recorded and we will post it on our social media channels and on our websites shortly. Stay on for another minute. Otherwise, thank you all for your attention.